We've said earlier that ATP is a critically important molecule in microbial cells and all cells because it's really the energy currency that the cell uses. It's important for us to remember that the metabolic processes that we're talking about today, cellular respiration and oxidative phosphorylation, and even fermentation, have a purpose for the cell, and the purpose is the production of ATP. Ultimately, all of these long series of reactions are happening in order to produce ATP to meet the energy needs of the cell. Remember, ATP is a middleman molecule. This is a molecule that the cell is going to use for the short-term storage of energy, or what we often refer to as potential energy, or PE. ATP, recall, is used in the cell by having its outermost phosphate group broken off and then put back on, and then broken off and then put back on, as the cell sort of shifts energy around. The cell is going to harvest energy from exergonic reactions, catabolic reactions, and it's going to harvest that energy by storing it in that phosphate bond as it puts the phosphate onto ADP to make ATP. Then, when an anabolic reaction begins to occur, when an endergonic reaction occurs, one that requires energy, the cell will simply take that outer phosphate bond off. It will break that bond and release the energy that's needed by that reaction. So ATP is the primary molecule for this very short-term management of energy. When the phosphate group is on the molecule, we've got a little bit of energy stored in there. When the phosphate group is taken back off, the energy is released. ATP is used to couple exergonic and endergonic reactions. Now, here's something we haven't said yet, but something that's very important. Cells do not store large amounts of ATP. They only store up to several minutes worth. Now that may seem strange. You would think that a molecule that is as important as ATP is would be kept in surplus in a cell, just in case a time arose when it wasn't able to make it. But in fact, cells are very thrifty, and they don't make anything they don't need in that moment. So ATP is not made and put away for a rainy day. ATP is made steadily over time as the cell has a need for the molecule. That tells us something. That tells us that microbial cells and other cells must be able to make ATP quite quickly whenever it's required. ATP is critical because ATP is this middleman in shifting energy around, in capturing it when it's released from one kind of reaction, and then providing it when it's needed for another kind of reaction. All cells do this kind of thing all the time. They have catabolic reactions going on over here, then they have anabolic reactions going on over here. Some reactions are releasing energy, some reactions need energy, and it's ATP that fills in that role. All cells need a constant, ongoing source of energy in order to do all the work they need to do every day. And as cells build molecules through anabolic processes, the electron pair that sits in every covalent bond is a source of stored energy. that pair of electrons has energy associated with them. But while they're sitting in a covalent bond, that energy is sitting with them. It is stored with them. But when a cell turns around and breaks a molecule, breaks the covalent bonds, those electrons are going to be released. And those electrons are then on the move. Remember, we said electrons are the conduits of energy. Just like a pipeline carries water, the electron carries energy with it. So electrons are traveling, if you will, an energy pathway in the cell from one bond to another bond. They're being released, and then they're being put back into some other molecule, or they're being transformed into a different form of energy. In a covalent bond, there's a pair of electrons, one from one atom and one from another atom. Now, most metabolic reactions, whether they're catabolic or anabolic, are these oxidation reduction type of reactions. Remember, we've said most metabolic reactions inside living cells are enzyme requiring reactions. And now we've added on to that not only do most biological reactions require enzymes, but they're also the oxidation reduction type. Now let's think a little bit more about what these words mean. Oxidation reduction. We can say now that an atom that loses an electron, is going to be oxidized. An atom that gains an electron is going to be reduced. And these two things always go together. So if something's being oxidized during a chemical reaction, something else is being reduced. Now, there are lots of little memory devices, mnemonic devices that people use to remember which is which when it comes to oxidation reduction. Which one is loss of electron, which one is gain of electron. The mnemonic device that I use is Leo goes grrr. Leo the lion goes grrr. And the way you use this device is that Leo stands for lose electron oxidation, and grrr stands for gain electron reduction. So you can use that little memory device to trigger your memory which reaction is oxidation and which is reduction. Just remember that oxidation always is paired with reduction. Those two things always go together. As we start talking about oxidation reduction and energy in electrons, we also have to remember the first law of thermodynamics. Remember, the law tells us that energy cannot be created in a cell. It cannot be destroyed in a cell. The only thing that can happen to it is it gets transformed from one type to another type. So as we watch electrons move from place to place, as we watch them be set free out of a bond, we're also going to see them being converted or altered or transformed from one kind of energy to another. Remember, we said we can describe the bonds in a molecule as chemical energy. That's a form of stored or potential energy. We said there's energy in movement. There's kinetic energy. There's energy that is related to heat. There's thermal energy. There are different forms of energy. And the cell can transform energy from one to another. as electrons are released and as they move from place to place in a cell, the energy that they carry with them can be used by the cell to do work. That's all energy is. It's the ability to do work. So we've said that cellular respiration is followed by oxidative phosphorylation, and that can be aerobic or anaerobic. And the purpose of those two sets of reactions is to produce ATP. We've also said that fermentation is a long series of reactions whose purpose is to make ATP. Now we need to think a little bit about how the ATP is made, the literal mechanism of how the ATP is made. And again, we've talked about this a little bit. Remember, we talked about substrate level phosphorylation. We said that there are some enzymes that perform catalysis during chemical reactions that have sort of an extra active site on them that allows for ATP to sort of be made during this piggyback reaction. As the enzyme takes a phosphorylated substrate, a substrate molecule that has a phosphate group attached to it, as the enzyme takes that substrate and turns it into product, along the way, sort of piggybacked on top of that, a phosphate group will be added onto ADP and make ATP. That's a process called substrate level phosphorylation. It turns out that there are times during cellular respiration when substrate level phosphorylation is going to occur, and a little bit of ATP is going to get made. Specifically, there's a process during cellular respiration called glycolysis, and a little substrate level phosphorylation happens during glycolysis. There's another set of reactions during cellular respiration called the Krebs cycle, and a little bit of ATP gets made during the Krebs cycle using this mechanism of substrate level phosphorylation. We also see substrate level phosphorylation occurring during fermentation. So it happens all over the place during these metabolic reactions. But remember, it's not a great way to make ATP if you're a living cell, and that's because it's inefficient. Every time the enzyme takes the substrate and creates product with it, it's also going to make one molecule of ATP. That's not a good way to make ATP. We need a ready, steady source of ATP, and substrate level phosphorylation is not the mechanism to do that. It's going to make some ATP, so, we'll take it. We'll take it as a living cell, but we need a better mechanism. We need a more efficient mechanism as well. And that's where oxidative phosphorylation comes in. Oxidative phosphorylation, remember, is a series of reactions that's going to happen after cellular respiration occurs. It's oxidative phosphorylation, as we said a minute ago, that's going to involve a hydrogen ion gradient. And the hydrogen ions are going to pass down their concentration gradient through that molecule called ATP synthase that's sitting in the cell membrane. And as the gradient goes away, as it resolves, as it goes back to equilibrium, the ATP synthase molecule is going to make ATP for us. And it does it very efficiently. For every molecule of glucose that gets made, or I should say that gets sent into cellular respiration and oxidative phosphorylation, for every molecule of glucose, we're going to get anywhere from 26 up to 30 or more ATP molecules made. That's a heck of a lot better than the one-to-one inefficient reaction that is substrate level phosphorylation. So let's take a minute again and summarize. I'm purposefully being repetitive today because you have to get comfortable with all of this very cumbersome terminology in order for us to master this material about microbial metabolism. So I'm purposefully saying these things again and again and again to try to make it so that you're more comfortable with the language of metabolism. So, some microbes are going to do cellular respiration and oxidative phosphorylation to make their ATP. If they're aerobic organisms, they're going to do it with oxygen. If they're anaerobic organisms, they're going to do it with some other molecule, like nitrate or sulfate or something else. The purpose of the cellular respiration and the oxidative phosphorylation is to make ATP, and to make it quite efficiently for the ongoing energy needs of the cell. Some microbes are going to do cellular respiration and oxidative phosphorylation as their primary preferred metabolic pathway to make ATP. But if necessary, they can also use fermentation. And if they use fermentation, they're going to still make some ATP, but they're going to make it through substrate level phosphorylation, and that's an inefficient way to do it. There are still other microbes who don't do cellular respiration and oxidative phosphorylation at all. There are some microbes who use fermentation as their preferred metabolic pathway. So they're doing fermentation and substrate level phosphorylation only. And they have evolved to be able to do that. They have evolved to be able to survive quite well, making their ATP in this very inefficient manner. Microbes are incredibly diverse in their metabolic abilities. You and I can't switch and swap from metabolic process to metabolic process. You and I can't do that. If we suddenly find ourselves without oxygen, if we find ourselves without the final electron acceptor that we need at the end of our oxidative phosphorylation pathway, we're in trouble. We've got maybe 4 or 6 minutes before we're dead. But microbes can switch and swap. Most of them are facultative. Most of them, if they don't have the electron acceptor they need, they can just switch and use a different one. They are incredibly diverse in their metabolic abilities.